Now, we will go in detail on the recombinant protein vaccine. A recombinant protein vaccine is a vaccine produced through recombinant DNA technology, similar concept on how the DNA vaccine is produced. This involves inserting the DNA encoding an antigen, which is a protein base, that stimulates an immune response. But the difference here is that the end product in this is a purified recombinant protein as a whole, as a vaccine antigen, instead of a purified plasmid DNA, like the one in the case of DNA vaccine. And this recombinant protein can be expressed in a heterologous expression systems like E. coli, yeast, or insect using the recombinant protein expression technology. So like I mentioned earlier, because subunit vaccine contain only the minimal component of the antigen and not all the other molecules that make up the whole pathogen, the chances of adverse reactions, particularly the autoimmune response to the vaccine, are lower. So this is one of the new advancement that has been made in the science, especially for vaccine production, which is to minimize the potential of autoimmune response when you are using only a minimal component of the vaccine instead of the whole pathogens as the vaccine antigens. So as you can see here, from the whole microorganisms, you can have a lot of multiple epitopes that can induce immune response. Then you can isolate to a specific minimal antigen, which is in this case is a protein. And still protein contains several epitopes. And from the proteins, it is still possible to isolate a really specific antigen in the peptide form, which is to induce a really specific immune response against particular peptide of the proteins. So along the way, as you can see, from A to B to C, the side effect is lesser and lesser. So one of the best examples of the recombinant protein vaccines that are currently in use is the vaccines against hepatitis B virus, which is HBV. So the current vaccine, the current one, the clinical use one for hepatitis B virus, is produced by expressing the HBV surface antigen using the yeast expression system. So as we can see here, first, the Hep B antigen producing gene is isolated from the hepatitis B virus by a normal isolation process like a mixture of cell lysis, protein denaturation, and also centrifugation. And then a plasmid is extracted from a bacterium and is cut with the restriction enzymes, as you can see here. The isolated Hep B antigen producing gene is located and inserted into the bacterial plasmid vector on forming the recombinant DNA. And this recombinant DNA containing the target gene is introduced into a yeast cell and further forming the recombinant yeast cell. And the recombinant yeast cell multiplies in the fermentation tank and produce the Hep B antigen, which is the protein that can induce the immune response as the vaccine antigen. And then from the fermentation tank, the mixture will be isolated and will be undergo extraction and purification of the HB vaccine, and the final purified Hep B antigen will be combined with preserving agents and other excipients to formulate a final vaccine formulation. So a more recently developed example of a recombinant protein vaccines is against HPV, which is the human papilloma virus. So basically, there are two vaccines, which are Gardasil and Cervarix. This is the commercial name, were approved for clinical use against HPV. But here, it's still produced based on recombinant protein, but they will form a virus-like particles morphology for this particular vaccine. So this is a general demonstration, representative diagram on how HPV vaccine is produced. So basically the HPV vaccine is based on L1 protein, which is the major capsid protein of human papilloma virus, and L1 is a protein with the ability to spontaneously self-assemble into virus-like particles. So first, we need to identify the L1 coding regions, and these L1 coding regions will be inserted into the plasmid DNA, and from the yeast, similar to how the Hep B vaccine produced, we will isolate and purify the L1 molecule that we produce, and this L1 molecule will be allowed to self-assemble in a system to produce the virus-like particles, and which mimic the native organization of a virus. Thus, is expected to induce a strong immune response. So this is the final formulation of Gardasil, where, as you can see here, it consists of 4 strains of human papillomavirus, which is strain 6, 11, 16, and 18, which is accountable for most of the cervical cancer case. So in each of these strains, they isolate the L1 and make them into self-assembled into virus-like particles, and the vaccines contain the cocktail of the virus-like particles that's coming from all of these strains based on L1 protein. And as you can see here, it is also adjuvanted by the alum in these vaccine formulations. So what do it means by the virus-like particles is basically they are a multi-protein structure that mimic the organization and conformation of authentic native virus but lack the viral genome. Means that it cannot give you illness, they cannot replicate, it's just mimicking the structural organization of the virus and without the viral genomes, thus yielding a safer and maybe cost-effective vaccine candidates.